Can we switch our brains on and off? Not really. Your brain is always doing something, even if you're fast asleep. But thanks to a brilliant mashup of brain cells and algae and fiber optic cable, we can throw a switch on specific bits of the brain. Neuroscience in the last few years has gone through a revolution. It's been a fairly quiet revolution. There hasn't been a lot of flag waving or guillotining, but the way that a lot of us study brain cells and networks in the brain has changed forever. The new technique, which is a hot tip for a Nobel in a few years' time, is called optogenetics, which is kind of a clunky name, but what it means is that we can use genes to control brain cells with light. That's right, in labs all around the world right now, neuroscientists are turning brain cells on and off using tiny beams of light. This is pretty awesome because up until now, the best way that we could make brain cells fire was by sticking in an electrode somewhere in the brain, connecting that to a power source and throwing the switch. That's a blunt tool because it's very hard to know how much of the brain and what type of brain cells you're turning on. So how does optogenetics work? Well, when a brain cell, a neuron, fires, lots of little channels open up on its surface. These channels let loads of charged ions into and then out of the cell. And this sets off a wave of charge that flows all the way down the neuron's fibers and sends the signal to other neurons. Now, what some scientists did about 9 years ago in Stanford was pinch an ion channel from an algae. This ion channel opens when it's hit with light. So the algae would normally use that channel to swim around and find the right light conditions for it to grow. But if we take the gene for that channel and put it into a neuron, and then shine light on the neuron, it sets off the same wave of charge. It makes the brain cell fire. Or if we use a different type of channel that sends charge in the other direction, we can stop brain cells firing. So by installing the genes for different types of channel in different types of brain cell, we can turn specific circuits in the brain on and off using different colors of light. It's as if the brain is a massive orchestra and the conductor just has a bunch of different colored laser pointers, and he can go, you, stop, and now you, start. Good, keep going, stop, and now you. This is really exciting, not just for neuroscientists, but for all of us. We can start to figure out how specific types of brain cell, in particular bits of the brain, affect what we do and who we are, and even how the brain goes wrong when it's not working properly. Already just this year, a group of scientists in Japan implanted a fiber optic cable in the brain of a mouse and shone a light to shut down a specific type of cell, and the mouse went to sleep. Another team in the US turned on a light inside a rat's brain and stopped epileptic seizures just by shutting down a different type of brain cell. Others have found that if you stimulate a particular circuit, you can tip mice into obsessive grooming, a bit like OCD. Or if you shut down another type of brain cell, you can help them get over cocaine addiction. It's a new era, it's incredibly powerful, and one day we might even see people treated with optogenetics. It won't be easy because first, we have to get the genes into the right brain cells, which will probably mean injecting a virus. And then we'll have to insert a fiber optic cable to turn the cells on or off. But if we can get it to work, it'll be incredibly precise, especially if you compare it to sticking in an electrode or bathing the whole brain in drugs, which is basically what we're left with at the moment in a lot of situations. So, optogenetics, genetic light switches for the brain. Kind of a funny name, but a very bright future. So if this is flicking switches in your brain and you haven't done it already, subscribe to Head Squeeze.